In the previous chapter, we looked at the broad classification of living organisms under the system proposed by Whittaker in 1969, wherein he suggested the five kingdom classification, that is Monera, Protista, Fungi, Animalia, and Plantae. In this chapter, we will deal in detail with further classification within kingdom Plantae, popularly known as plant kingdom. We must stress here that our understanding of what plant kingdom has changed over time. Fungi and member of Monera and Protista have now been excluded from Plantae through their earlier classification placed them in the same kingdom. So the cyanobacteria that are also referred to as blue-green algae are not algae anymore. In this chapter, we will describe algae, bryophyte, pteridophyte, gymnosperm and angiosperm under Plantae. Let us look at classification within the angiosperm to understand some of the concerns that influence the classification system. The earliest classification system used only gross superficial morphological characters such as habitat, color, number and shape of leaves, etc. They were based mainly on vegetative character or on the androecium structure system given by Linnaeus. Such systems were artificial. They separated the closely related species since they were based on a few characters. Also, the artificial system give equal weightage to vegetative and sexual character. This is not acceptable since we know that often the vegetative character are more easily affected by the environment. Against this, natural classification system developed, which was based on natural affinity among the organisms and considered not only the external feature but also internal feature like ultrastructure, anatomy, embryology and phytochemistry. Such a classification of flowering plants was given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification system based on evolutionary relationship between the various organisms are acceptable. This assumes that organisms belonging to the same taxa have a common ancestor. We now use information from many other sources to help resolve difficulties in classification. These become more important when there is no supporting fossil evidence. Numerical taxonomy, which is now easily carried out using computers, is based on all observable characteristics. Number and codes are assigned to all the characters and the data are then processed. In this way, each character is given equal importance and at the same time hundreds of characters can be considered. Cytotaxonomy, that is based on cytological information like chromosome number, structure, behavior, and chemotaxonomy, that use the chemical constituent of the plant to resolve the confusions, are also used by taxonomists these days. 3.1 Algae. Algae are chlorophyll-bearing simple thalloid, autotrophic and largely aquatic, both freshwater and marine organisms. They occur in a variety of other habitats, moist stones, soil and wood. Some of them also occur in association with fungi, example lichen, and animal, example on a sloth bear. The form and size of algae is highly variable, ranging from colonial form like volvox and the filamentous form like Eulothrix and Spirogyra. A few of the marine forms such as kelp form massive plant bodies. The algae reproduce by vegetative, asexual and sexual method. Vegetative reproduction is by fragmentation. Each fragment develops into a thallus. Asexual reproduction is the production of different types of spores, the most common being the zoospore. They are flagellated, motile, and on germination give rise to new plants. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated and similar in size as in Eulothrix, or non-flagellated, non-motile but similar in size as in Spirogyra. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size as in species of Eudorhina is termed as an isogamous. Fusion between one large, one motile static female gamete and a smaller motile male gamete is termed oogamous. Example volvox and fucus. Algae are useful to man in a variety of ways. At least a half of the total carbon dioxide fixation on Earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. They are of paramount importance as primary producer of energy-rich compounds which form the basis of the food cycle of all aquatic animals. Many species of Porphyra, Laminaria and Sargassum are among the 70 species of marine algae used as food. Certain marine brown and red algae produce large amount of hydrocolloids, water-holding substances, example alzine, produced by brown algae, and kerosene produced by red algae, which are used commercially. Agar, one of the commercial products obtained from Galidium and Gracilaria, which are red algae, are used to grow microbes and in preparation of ice creams and jellies. Chlorella, a unicellular algae rich in proteins, is used as food supplement even by space travelers. The algae are divided into three main classes: Chlorophyceae, Phaeophyceae and Rhodophyceae. 3.1.1 Chlorophyceae. The members of Chlorophyceae are commonly called green algae. The plant body may be unicellular, colonial or filamentous. They are usually gross green due to the dominance of the pigment chlorophyll A and B. The pigments are localized in definite chloroplasts. The chloroplast may be discoid, plate-like, reticulate, cup-shaped, spiral or ribbon-shaped in different species. Most of the members have one or more storage bodies called pyrenoids located in the chloroplast. Pyrenoids contain protein besides starch. Some algae may store food in the form of oil droplets. Green algae usually have a rigid cell wall made up of inner layer of cellulose and an outer layer of pectose. Vegetative reproduction usually takes place by fragmentation or by formation of different types of spores. Asexual reproduction is by flagellated zoospores produced by Zoosporangia. The sexual reproduction saw considerable variation in the type and formation of sex cells, and it may be isogamous, anisogamous or oogamous. Some commonly found algae are Chlamydomonas, Volvox, Eulothrix, Spirogyra and Chara. 3.1.2 Phaeophyceae, that is brown algae. The members of Phaeophyceae or brown algae are found primarily in marine habitat. They show great variation in size and form. They range from simple branched filamentous form, example Ectocarpus, to profusely branched form as represented by kelp, which may reach a height of 100 meters. They possess chlorophyll A and C, carotenoids and xanthophyll. They vary in color from olive green to various shades of brown, depending upon the amount of xanthophyll pigment fucoxanthin present in them. Food is stored as complex carbohydrate, which may be in the form of laminaria or mannitol. The vegetative cell has a cellulosic wall, usually covered on the outer side by a gelatinous coating of algae. The protoplast consists, in addition to plastid, a centrally located vacuole and nucleus. The plant body is usually attached to the substratum by a holdfast and has a stalk, the stripe, and leaf-like photosynthetic organ, the frond. Vegetative reproduction takes place by fragmentation. Asexual reproduction in most brown algae is by biflagellated zoospores that are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous, anisogamous or oogamous. Union of gametes may take place in water or within the oogonium, oogamous species. The gametes are pyriform, that is pear-shaped, and bear two laterally attached flagella. The common form are Ectocarpus, Dictyota, Laminaria, Sargassum and Fucus. 3.1.3 Rhodophyceae, that is red algae. The members of Rhodophyceae are commonly called red algae because of the predominance of the red pigment R-phycoerythrin in their body. Majority of the red algae are marine with greater concentration found in warmer areas. They occur in both well-lighted regions close to the surface of water and also at great depth in ocean where relatively lighter light penetrates. The red thalli of most of the red algae are multicellular. Some of them have complex body organization. The food is stored as Floridian starch, which is very similar to amylopectin and glycogen in structure. Red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spores and sexually by non-motile gametes. Sexual reproduction is oogamous and accomplished by complex post-fertilization development. The common members are Polysiphonia, Porphyra, Galidium, and Gracilaria. Bryophytes 3.2. Bryophytes include the various mosses and liverworts that are found commonly growing in moist shade areas in hills. Bryophytes are also called amphibians of the plant kingdom because these plants can live in soil but are dependent on water for sexual reproduction. They usually occur in damp, humid, and shaded localities. They play an important role in plant succession on bare rock or soil. The plant body of bryophyte is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by unicellular or multicellular rhizoids. They lack true root, stem, or leaves. They may possess root-like, leaf-like, or stem-like structure. The main plant body of the bryophyte is haploid. It produces gametes, hence is called gametophyte. The sex organ in bryophytes are multicellular. The male sex organ is called antheridium. They produce biflagellated antherozoids. The female sex organ is called archegonium, is flask-shaped and produce a single egg. The antherozoids are released into water where they come in contact with archegonium. An antherozoid fuses with the egg to produce the zygote. The zygote do not undergo reduction division immediately. They produce a multicellular body called a sporophyte. The sporophyte is not free-living but attached to the photosynthetic gametophyte and derive nourishment from it. Some cells of the gametophyte and sporophyte undergo reduction division, meiosis, to produce haploid spores. These spores germinate to produce gametophyte. Bryophytes in general are of little economic importance, but some mosses provide food for herbaceous mammals, birds, and other animals. Species of sphagnum, a moss, provide peat that have long been used as fuel and as packing material for transshipment of living materials because of their capacity to hold water. Mosses, along with lichen, are the first organism to colonize rocks and hence are of great ecological importance. They decompose rocks, making the substrate suitable for the growth of higher plants. Since mosses form dense mats on the soil, they reduce the impact of falling rain and prevent soil erosion. The bryophytes are divided into liverworts and mosses. 3.2.1 Liverworts. The liverworts grow usually in moist, shady habitats such as bank of streams, marshy ground, damp soil, bark of tree, and deep in the woods. The plant body of liverwort is thalloid. Example of liverwort is Markensia. The thallus is dorsiventrally and closely pressed to the substrate. The leafy members have tiny leaf-like appendages in two rows on the stem-like structures. Asexual reproduction in liverworts takes place by fragmentation of thalli or by the formation of a specialized structure called gamy, single gamma. Gamy are green multicellular asexual buds which develop into small receptacles called gamma cup located on the thalli. The gamy become detached from the parent body and germinate to form new individual. During sexual reproduction, male and female sex organs are produced either on the same or on different thalli. The sporophyte is differentiated into a foot, seta, and capsule. After meiosis, spores are produced within the capsule. These spores germinate to form free-living gametophytes. 3.2.2 Mosses. The predominant stage in the life cycle of moss is the gametophyte, which consists of two stages. The first stage is the protonema stage, which develops directly from the spore. It is a creeping, green, branched, and frequently filamentous stage. The second stage is the leafy stage, which develops from the secondary protonema as a lateral bud. They consist of upright cylinder axis bearing a spirally arranged leaves. They are attached to the soil through multicellular and branched rhizoids. This stage bears the sex organs. Vegetative reproduction in mosses is by fragmentation and budding in the secondary protonema. The sexual reproduction and the sex organs, antheridia and archegonia, are produced at the apex of the leafy shoot. After fertilization, the zygote develop into the sporophyte, consisting of a foot, seta, and capsule. The sporophyte in mosses is more elaborate than that in liverworts. The capsule contains spores. Spores are formed after meiosis. The mosses have an elaborate mechanism of spore dispersal. Common examples of mosses are Funaria, Polytrichum, and Sphagnum, that is peat. 3.3 Pteridophytes. The pteridophytes include Horstella and fern. Pteridophytes are used for medicinal purposes and as soil binders. They are also frequently grown as ornamentals. Evolutionarily, they are the first terrestrial plant to possess vascular tissues, xylem, and phloem. You shall study more about these tissues in chapter 6. The pteridophytes are found in cool, damp, and shady places, though some may flourish well in sandy soil conditions. You may recall that in bryophytes, the dominant phase in the life cycle is the gametophytic plant body. However, in pteridophytes, the main plant body is a sporophyte, which is differentiated into true root, stem, and leaves. These organs possess well-differentiated vascular tissues. The leaves in pteridophytes are small, microphyllous, as in Selaginella, or large, macrophyllous, as in fern. The sporophytes bear sporangia that are subtended by leaf-like appendages called sporophyll. In some cases, a sporophyll may form distinct compact structure called strobili or cone, Selaginella and equisetum. The sporangia produce spores by meiosis in a spore mother cell. The spores germinate to give rise to inconspicuous small but multicellular, free-living, mostly photosynthetic thalloid gametophyte called prothallus. These gametophytes require cool, damp, shady places to grow. Because of this specific restricted requirement and the need for water for fertilization, and the spread of living pteridophytes is limited and restricted to a narrow geographical region. The gametophytes bear male and female sex organs called antheridia and archegonia respectively. Water is required for transfer of antherozoids, the male gamete released from the antheridia to the mouth of archegonium. Fusion of male gamete with the egg present in the archegonium result in the formation of zygote. Zygote thereafter produce a multicellular, well-differentiated sporophyte, which is the dominant phase of the pteridophyte. In majority of the pteridophytes, all the spores are of a similar kind. Such plants are called homosporous. Genera like Selaginella and Salvinia, which produce two kinds of spores, macro large and micro small spores, are known as heterosporous. The megaspores and microspores germinate and give rise to female and male gametophyte respectively. The female gametophyte in these plants are retained in the parent sporophyte for variable periods. The development of the zygote into young embryo takes place within the female gametophyte. This event is a precursor to the seed habit considered an important step in evolution. The pteridophytes are further classified into four classes: Psilopsida, Xylotum, Lycopsida, Selaginella and Lycopodium, Sphenopsida, Equisetum, and Pteropsida, Dryopteris, Pteris, and Adiatum. 3.4 Gymnosperm. The gymnosperm, gymno means naked, sperma means seed, are plant in which the ovules are not enclosed in any ovary wall and remain exposed both before and after fertilization. The seed that develop post-fertilization are not covered, that is, are naked. Gymnosperm include medium-sized trees or tall trees and shrubs. One of the gymnosperm, the Ncert redwood tree sequoia, is one of the tallest tree species. The roots are generally taproot. Roots in some genera have fungal association in the form of mycorrhiza pinus, while in some other cycas, small specialized roots called caroloid roots are associated with N2-fixing cyanobacteria. The stem are unbranched cycas or branched pinus and cedrus. The leaves may be simple or compound. In cycas, the pinnate leaf persists for a few years. The leaves in gymnosperm are well adapted to withstand extreme temperature, humidity, and wind. In conifers, the needle-like leaf reduce the surface area. Their thick cuticle and sunken stomata also help to reduce water loss. The gymnosperm are heterosporous. They produce haploid microspores and megaspores. The two kinds of spores are produced within sporangia that are borne on a sporophyll, which are arranged spirally along an axis to form lax or compact strobili or cone. The strobili-bearing microsporophyll and microsporangia are called microsporangiate or male strobili. The microspores develop into a male gametophyte generation, which is highly reduced and is confined to only a limited number of cells. This reduced gametophyte is called pollen grain. The development of pollen grain takes place within the microsporangia. The cones bearing megasporophyll with ovule or megasporangia are called macrosporangiate or female strobili. The male or female cones or strobili may be born on the same tree, example pinus. However, in cycas, male cone and megasporophyll are born on different trees. The megaspore mother cell is differentiated from one of the cells of the nucleus. The nucleus is protected by envelopes and the composite structure is called ovule. The ovule are born on megasporophyll, which may be clustered to form the female cone. The megaspore mother cell divided meiotically to form 4 megaspores. One of the megaspores enclosed within the megasporangium develops into the multicellular female gametophyte that bear 2 or more archegonium or female sex organ. The multicellular female gametophyte is also retained within the megasporangium. Unlike bryophytes and pteridophytes, in gymnosperm the male and female gametophyte do not have an independent free-living existence. They remain within the sporangium. Retained on the sporophyte, the pollen grain is reduced from the microsporangium. The pollen grain is released from the microsporangium. They are carried in air currents and came in contact with the opening of the ovules born on megasporophyll. The pollen tube carrying the male gamete grow towards archegonia in the ovule and discharge their content near the mouth of the archegonia. Following fertilization, zygote develop into embryo and the ovule into seed. These seeds are not covered. 3.5 Angiosperm. Unlike the gymnosperm where the ovule are naked, In the angiosperm or flowering plant, the pollen grains and ovule are developed in a specialized structure called flowers. In angiosperm, the seeds are enclosed in fruits. The angiosperms are an exceptionally large group of plants occurring in a wide range of habitat. They range in size from the smallest wolfia to tallest tree of eucalyptus over 100 meters. They provide us with food, fodder, fuel, medicine, and several other commercially important products. They are divided into two classes, the dicotyledony and the monocotyledony. The dicotyledons are characterized by seeds having two cotyledons, reticulate venation in leaves, and tetramerous or pentamerous flowers, that is having 4 or 5 members in each floral whorls. The monocotyledons, on the other hand, are characterized by single cotyledonous seeds, parallel venation in leaves, and trimerous flower having 3 members in each floral whorls. The male sex organ in flower is the stem. Each stamen consists of a cylinder filament with another at the tip. Within the anthers, the pollen mother cell divides by meiosis to produce microspores which matures into pollen grain. The female sex organ in a flower is the pistil. Pistil consists of a swollen ovary at its base, a long cylinder, style, and stigma. Inside the ovary, ovules are present. Generally, each ovule has a megaspore mother cell that undergoes meiosis to form 4 haploid megaspores. 3 of them degenerate and 1 divide to form the embryo sac. Each embryo sac has 3 celled egg apparatus, 1 egg cell, 2 synergids, and 3 antipodal cells, and 2 polar nuclei. The polar nuclei eventually fuse to produce a diploid secondary nucleus. Pollen grain, after dispersal from the anther, are carried in wind or various other agents to the stigma of a pistil. This is termed as pollination. Pollen grains germinate into the stigma and resulting pollen tube grow through the tissues of stigma and style and reach the ovule. The pollen tube enters the embryo sac where 2 male gametes are discharged. One of the male gametes fuses with the egg cell syngamy to form the zygote. Other male gamete fuse with the diploid secondary nucleus to produce the triploid primary endosperm nucleus (PEN) Ncert. Because of the occurrence of 2 fusions, that is syngamy and triple fusion, this event is termed as double fertilization, an event unique to angiosperms. The zygote develops into an embryo with 1 or 2 cotyledons, and the pen develops into endosperm which provide nourishment to the developing embryo. The synergid and antipodals degenerate after fertilization. During these events, the ovule develop into seed and ovaries develop into fruit. The life cycle of an angiosperm is shown in figure 3.6. Plant life cycle and alternation of generation. In plants, both haploid and diploid cells can divide by mitosis. This ability leads to the formation of different plant bodies, haploid and diploid. The haploid plant body produces gametes by mitosis. This plant body represents a gametophyte. Following fertilization, the zygote also develops by mitosis to produce a diploid sporophytic plant body. Haploid spores are produced by this plant body by meiosis. These in turn divided by mitosis to form a haploid plant body once again. Thus, during the life cycle of any sexually reproducing plant, there is an alteration of generation between gamete-producing haploid gametophyte and spore-producing diploid sporophyte. However, different plant groups as well as individual representing them different in the following patterns. First is saprophytic generation is represented only by the one-celled zygote. There is no free-living sporophyte. Meiosis in the zygote results in the formation of haploid spores. The haploid spores divide mitotically and form a gametophyte. The dominant photosynthetic phase in such plants is the free-living gametophyte. This kind of life cycle is termed as haplontic. Many algae such as volvox, Spirogyra and some species of Chlamydomonas represent this pattern. Second one is, on the other extreme, is a type where the diploid sporophyte is the dominant phase, photosynthetic independent phase of the plant. The gametophytic phase is represented by a single to few-celled haploid gametophyte. This kind of life cycle, termed as diplontic, an algae Fucus species represent this pattern. In addition, all seed-bearing plants, that is gymnosperm and angiosperm, follow this pattern with some variations, where the gametophytic phase in few to multi-celled. Bryophytes and pteridophytes, interestingly, exhibit an intermediate condition, haplodiplontic. Both phases are multicellular, however, they differ in their dominant phase. A dominant independent photosynthetic thalloid or erect phase is represented by a haploid gametophyte, and it alternates with the short-lived multicellular sporophyte, totally or partially dependent on the gametophyte for its anchorage and nutrition. All bryophytes represent this pattern. The diploid sporophyte is represented by a dominant, independent photosynthetic vascular plant body. It alternates with the multicellular, saprophytic, autotrophic, independent but short-lived haploid gametophyte. Such a pattern is known as haplodiplontic life cycle. All pteridophytes exhibit this pattern. Interestingly, while most alga genera are haplontic, some of them such as Ictocarpus, Polysiphonia, kelp are haplodiplontic. Fucus, an algae, is diplontic. Summary of the chapter. Plant kingdom includes algae, bryophyte, pteridophyte, gymnosperm and angiosperm. Algae are chlorophyll-bearing simple thalloid, autotrophic and largely aquatic organisms. Depending on the type of pigment possessed and the type of stored food, algae are classified into three classes, namely chlorophyci, phaeophyci and rhodophyci. Algae usually reproduce vegetatively by fragmentation, asexually by formation of different types of spores, and sexually by formation of gametes which may show isogamy, anisogamy or oogamy. Bryophytes are plants which can live in soil but are dependent on water for sexual reproduction. Their plant body is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by rhizoids. They possess root-like, leaf-like and stem-like structure. The bryophytes are divided into liverwort and mosses. The plant body of liverwort is thalloid and dorsiventral, whereas mosses have upright cylinder axis bearing spirally arranged leaves. The male plant body of bryophyte is gamete-producing and is called a gametophyte. It bears the male sex organ called antheridia and the female sex organ called archegonia. The male and female gametes fuse to form zygote. which produces a multicellular body called a sporophyte. It produces haploid spores. The spores germinate to form gametophyte. In pteridophyte, the main plant body is saprophyte, which is differentiated into true root, stem, and leaf. These organs possess well-differentiated vascular tissue. The sporophyte bear sporangia which produce spores. The spores germinate to form gametophyte which require cool, damp places to grow. The gametophyte bear male and female sex organs called antheridia and archegonia respectively. Water is required for transfer of male gamete to archegonium, where zygote is formed after fertilization. The zygote produces a sporophyte. The gymnosperm are the plant in which ovules are not enclosed by any ovary wall. After fertilization, the seed remain exposed, and therefore these plants are called naked seed plant. The gymnosperm produce microspores and megaspores, which are produced in microsporangia and megasporangia borne on the sporophyll. The sporophylls, microsporophylls, and megasporophylls are arranged spirally on axis to form male and female cones respectively. The pollen grains germinate and the pollen tube releases the male gamete into the ovule, where it fuses with the egg cell in archegonia. Following fertilization, the zygote develops into embryo and the ovule into seed. In angiosperm, the male sex organ stamen and female sex organ pistil are born in a flower. Each stamen consists of a filament and an anther. The anther produces pollen grains, male gametophyte. After meiosis, the pistil consists of an ovary enclosing one to many ovules. Within the ovule is the female gametophyte or embryo sac, which contains the egg cell. The pollen tube enters the embryo sac, where two male gametes are discharged. One male gamete fuses with egg cell synagamy and other fuse with diploid secondary nucleus. Triple fusion. This phenomenon of two fusion is called double fertilization and is unique to angiosperms. The angiosperms are divided into two classes, the dicotyledonous and the monocotyledonous. During the life cycle of any sexually reproducing plant, there is alteration of generation between gamete-producing haploid gametophyte and spore-producing diploid sporophyte. However, different plant groups as well as individuals may show different patterns in life cycle, haplontic, diplontic, or intermediate. Thank you very much for listening to this audiobook. Please do subscribe the channel and like and share the video.